a reagents company, and we're based in Cambridge in the UK, and we have a sales office in St. Louis in Missouri. And we provide a range of services and products primarily aimed at stem cell science and related areas. So the product that we're here to talk about today is LiveLight, and this is a product that has been developed specifically to address the problems associated with phototoxicity. So namely, when you're working with cells which are sensitive to light, either light for prolonged periods of low light or short periods of intense light. And this can have quite a dramatic impact on the survival of cells and therefore the data that you generate with your cells. So I'm going to pass over at this point to one of our presenters, Dr. Sami Barmada, who is a clinical neurologist at the University of Michigan in Ann Arbor. And he's been doing research on various neuronal diseases, and he's going to share some of his experiences with using LiveLight. And then subsequent to that, if I can get Dr. John Stockley back on the call, he'll share with you a bit more insight into how the LiveLight system works. So if I may, Sami, I will hand it over to you.
Okay, so hello. Sorry for the delay. I'm hoping that everyone can, nope, I'm still seeing John. I'm hoping that everyone can see the presentation right now. And welcome, my name is Sami Barmada. I am a neurologist, as Michael mentioned, but I spent a lot of my time doing research on neurodegenerative diseases, so ALS and different types of dementia. And imaging, so we do a lot of our experiments in neurons, either derived from human stem cells or from primary rodent neurons. And the use of LiveLight has, I think, changed our approach somewhat. It's helped quite a bit in that, as you heard Michael say, that LiveLight was engineered or created specifically to maximize the survival of cells in an environment in which they're illuminated by potentially toxic wavelengths of light. And that's what we do. We do a lot of fluorescence microscopy, and the phenotype that we're interested in is neurodegeneration, right? Is the death of neurons. And so we use a technique or a technology that was pioneered by Steve Finkbeiner at the Gladstone Institute that allows us to actually follow prospectively individual neurons over time, right? So we're interested in this phenotype of cell death, so we want to track it. So you can see here that cells do clearly die over time. And so the system enables us to go back again and again to the exact same cells in a population for thousands of cells. And so we can really detect the events or some sort of physiologic process that is most tightly connected with cell survival or cell death. And so this is this advantage of using a longitudinal analysis rather than a snapshot approach, because we can, as shown here, we can say, okay, there's this cell here outlined in the red that dies fairly quickly, and the other cells that live a little bit longer, and the cell outlined in the blue, which lives the entire length of the experiment. And so we want to know, what is associated with cell health or cell death. And so in that respect, it really helps to have, a relevant disease model in which your cells are not dying because of the media that they're in, for instance, or because of extraneous stimuli like light. So the basis of the system, we take multiple images. They're high resolution images, and so they're relatively small, so we don't see very much, but then we can stitch them together to get a fair representation of each well, and we can do this in a semi-high throughput fashion for, say 96 well plate. And then once we have those images representing the well of a plate with, maybe 100 or so neurons or more, then we've designed the algorithms that go and look at the image-based data. So the quality of the image is key, and I'll get back to that a little bit later. But we go back to the image-based data and try to segment and identify what are neurons in this population. And we then outline the neuron, the perimeter of the neuron, and assign an identifying number. And as I mentioned, we can come back again and again and again to the same cells. And then the program will actually go through over time and determine when each cell dies and then spit out a time of death. And so we can watch this process of neurodegeneration in real time. So the output from these experiments is survival data. So we can tell how long cells in a specific population live and what % are alive at any given time. This is Kaplan-Meier survival analysis, just like you see in a human epidemiological study. And more often we actually show the cumulative risk of death, which is really the same data. It's instead of looking at the % survival on the y-axis, you're looking at an exponential function, which is the probability of cell death, or the cumulative probability of cell death. Okay, so let's get back to imaging with LiveLight media. So this is these are two wells. One had LiveLight pneumo with SOS, and the other one had the media that we typically use, which is neurobasal media. And just looking by eye, they don't look that much different. Perhaps there are a few more neurons in the LiveLight. So let me back up and say that these are transiently transfected neurons that express a constitutively expressed fluorescent protein, in this case M-APL. And so you can see quite a bit more, I think, morphology with LiveLight than you can with neurobasal, but it's hard to tell at this magnification. But we use this to determine survival. How well do these cells do over time? And so in the longitudinal analysis, we compared neurobasal media to pneumo SOS, and then somebody in the lab thought that maybe it was the presence of thyroid hormones in pneumo and SOS that actually enhanced survival, and so we added extra thyroid T3, T4 hormones to neurobasal media. And so the reference population here is in gray, and you can see that using pneumo SOS LiveLight media, we have a reduction in the risk of death. So HR is hazard ratio, and that's a relative risk of death. So we reduce the risk of death by about 15%. And that's a highly significant, at least by the statistical test we use here, is this Cox proportional hazards analysis. And then, interestingly, adding thyroid hormone actually made things worse, so we don't do that anymore. But this is clear evidence that using pneumo LiveLight media actually does prevent some cell death that might be due to light or other factors in the media. And you can see here we have lots of neurons. We have about 2,500 neurons per population, so that's fairly good size, good powered study. All right, so we don't just look at survival. We look at lots of phenotypes in the cells, and one of the very fun toys that we've had to work with are photoconvertible proteins. And this is one example. This is Dendra2, expressed in a primary neuron, and it is a green fluorescent protein. But if you illuminate it with low wavelength light, in this case 405 nanometer light, you can get that protein to irreversibly convert to a red fluorescent state. Now the key here is that we're using low wavelength light, 405 nanometers, which prolonged exposure with that sort of wavelength light does not do good things to cells. So we know from experience that you can really kill your cells by illuminating with this low wavelength light. And so what we did was, in a photoconversion experiment where we illuminated cells with a lot of this low wave light, we looked to see if pneumo would provide any additional benefit, and it did. So in this case, when we did a photoconversion experiment in neurobasal in the gray and pneumo and SOS, this time we saw a hazard ratio of 0.69, so that's about a 30% reduction in the risk of death. That's fantastic. For our experiments, that makes a big, big difference. All right, so let's get back to the imaging characteristics of pneumo, of LiveLight media. So I showed this picture before. This is the raw data from the experiments, and if we contrast the image from the LiveLight media appropriately, this is what we get. It looks really beautiful. We can see a lot of processes, a lot of detail. If we use the same brightness contrast settings and apply those to the neurobasal media, this is what we get. So really a very, I think, valuable addition to LiveLight is it reduces background fluorescence. And that's what we're really seeing in the neurobasal media, is background fluorescence from the media. Now, if you look at the higher magnification, actually we just zoomed in on some of the cells. On the left is pneumo SOS, on the right is neurobasal, and when you contrast them appropriately and individually for the conditions in which they're cultured, they look pretty similar. Maybe a few, maybe a little bit better with pneumo, but we know what it looks like when you contrast them the same. In this case, we wanted to compare the intensity, the fluorescence intensity that we get. And so down at the bottom here, you can see average GFP intensity from, on the left are the 2,500 neurons in each population. You can see that actually we're getting, on average, higher fluorescence intensity that we measure from pneumo SOS. So in addition to the lower background, I think we're seeing an enhanced signal in transfected cells. And we also did the same experiment on the right. That bar graph is actually in cells transfected with Dendra2, so they have an overall lower intensity than GFP transfected cells. But still, in pneumo SOS, there's a greater signal in these cells. So it does help. And then when we look at our ability to distinguish specific features, and neurons are great because they have these very long, thin processes that we need to be able to resolve with high accuracy if we're going to be able to determine cell survival. In this case, what we did was we drew a line, and then we looked at the pixel intensity over the distance of that line, and that's what this little plot is on top. Many of you are familiar with ImageJ or Fiji. This is just a plot of intensity over distance, and it shows you that for each process there's a spike, right? And so this is a pneumo, and it's very easy to distinguish spikes from the background, right? So we can easily tell processes. When we do that same thing in neurobasal media, we can see that the spikes are not, they're relatively close to the background intensity level. And so you can see I've highlighted here in the red, these are the intensity values for the axonal processes, or dendrites, sorry, of that neuron, and you can see that it's fairly close to background. So the signal-to-noise ratio is much greater in pneumo plus SOS, which allows us to, again, identify these neurons with higher accuracy. It's very important to us, right? Because if you have an image like this, we need to be able to tell what's a neuron, what's not a neuron, and then to accurately draw a perimeter around those cells, because we want to measure the morphology of the neuron, its arborization, and also some intracellular processes such as, if we can, activity. And more so, this is a dynamic thing. So it's not just we want to do it at one time. It has to be every time. It has to be very consistent. And this allows us to say exactly when that cell dies. And this is just another example of that. These cells can change a lot over time, and so we need to have that high signal-to-noise ratio. And that, I think, is really what LiveLight has helped us to achieve. All right, and these are just some examples. So I was showing you single color fluorescence imaging before. This is dual color fluorescence imaging. This is in the red, the RFP and the GFP channels in one of our disease models. And again, you can just see that the quality of the images is very high. This is only a 20x high numerical aperture objective, and yet we're seeing quite a bit of detail, and that's very valuable. This is another example of a similar experiment in which we're able to use a live nuclear stain also in addition to the other channels. And so this is a 3-color image, again showing us high detail that we can use to pick out neurons. And then lastly, this is a pretty experiment which I false colored some of the cells. But if you look at high, again, we just zoomed in. This is a 20x objective, and yet we're seeing some features sort of pointed out here with the arrows, like dendritic spines, that we wouldn't be able to pick out if we weren't using pneumo and SOS. So I think it's helped us to, in 2 ways, really. The first is to, we don't have to worry about toxicity arising from the media or from light. We can focus on disease-specific toxicity, these genes that we think are important for causing neurodegeneration. We can actually look at the relevant physiologic pathways. And then second is the image quality. Right? It actually plays a large part in our experiments, being able to identify cells and their features. And that's really all I had today. I think I'm happy to take questions probably afterwards. I think John has actually joined the conversation. I may hand it back over to Michael right now so that he can introduce John.
Okay, thank you very much, Sami. Yeah, so we'll leave any questions that anybody might have until John has talked. So I'm glad to tell you that John has been able to join us. John is the person that actually led the discoveries that have resulted in the formulation of the LiveLight media. So this is work that John did while he was working at the University of Cambridge, and he's now working at MedImmune just down the road. And John, while he was working at Cambridge, he was working on oligodendrocyte precursor cells, and I think it was largely his frustration with the available media that really led to the development of the LiveLight system. So I'll hand it over at this point to John, and he can take you through a few more of the technical aspects of the system and the way he's been using it with oligodendrocytes and other neuronal cells. Thanks, John.
Thanks, Michael. I hope you can hear me just fine. And if you can maybe let me know if this is showing up, is it? So
we can see that fine.
You can see that fine? Is that in full screen, so?
Yes, that's good.
All right, so thanks everyone for bearing with us while we had a few technical glitches. So I'm going to just try and speed through basically the creation and how we came about with the media formulations. And Sami's shown some excellent data, what you can do with it, and I'm going to show you some of the other applications you can do it. But also to kind of give yourself a bit of a, give a bit of thought to your experiments and as to why you'll also potentially need the media. So I'm going to just briefly cover that. Light is constantly being used in biology now, and there are a lot of common applications for these light tools and why they warrant attention. And I'll show you that all cells from the central nervous system are altered by light, and where we can identify the sources of damage that occur from photochemical reactions. It's in the media and it's also in the cells. And I'll show you that some evidence that LiveLight confers protection to cells even when it's not there, but when you are using LiveLight, you can possibly expose cells up to 20 times or more levels of light without any adverse side effects. So light is an expanding tool. We can use it to control cellular functions with techniques such as optogenetics, which uses light-sensitive proteins such as channel rhodopsin 2 or cryptochrome 2, melanopsin, or these light oxygen voltage sensitive proteins. As Sami was showing, you can do live cell imaging, but the amount of live cell imaging that we're doing today is constantly increasing. We have super resolution imaging. We can image ions or voltages in cells, and it's even been used for automation. And light can also be used to deliver drugs because you can have temporal and spatial control over the release of drugs if they're caged. And light has also been used for sorting cells. So there's 40 years of knowledge showing that even ambient light kills cells. And all of these papers show you that if there's anything that's going to happen, if you shine light onto a cell long enough, you're going to end up with essentially a dead cell. And the reasons for that are many. Now, we sought to figure out why cells die from light for two reasons. The first reason was that we wanted to perform optogenetic stimulation, that is using blue light to activate cells and simulate neuronal communication to them. And to do that, we had this LED plate made, which sits above a normal 6-well plate, and you can shine light on it, and we can control the amount of light that reaches cells using this external stimulator. Now, it's very important to know the dose of light, because just as with a drug, it's all about concentration and exposure. So 1 milliwatt per millimeter squared of continuous light is equal to 1 kilojoule per meter squared. Now these are just units, but these are the units I'm going to be referring to. And now this doesn't sound like an awful lot, but this is actually the amount of light that you'd use. It's about half the amount of light you'd use during a standard image acquisition on a standard microscope. So by using rat primary neurons of different ages, of 7 days in vitro or 21 days in vitro, what we found was that when we shine light on them to simulate optogenetic stimulation, which is a standard technique almost these days, we found that the young neurons completely died, and we used propydium iodide exclusion assay for this, whereas older mature cortical neurons were a bit more resilient. Now, there are other cells in these cultures of enriched cortical neurons. We also have astrocytes, and these astrocytes can be stained for a protein called GFAP. And what we saw was that the numbers of these astrocytes weren't changing when we shine light on them, but we did notice morphological changes, and by using a process called a Shull analysis, which is essentially tracing the structure of these cells and then drawing circles around them with increasing radii from the center of the cell, and then plotting them as a number of processes that have that cross each of those radii, you can figure out just how complex these cells are. So what we can see is that when we shine light onto these astrocytes, they lose a number of processes, and they also retract. But OPCs were one of our main interests because oligodendrocyte precursor cells, or OPCs for short, they generate all of the myelinating oligodendrocytes in the central nervous system. And in diseases such as multiple sclerosis, these oligodendrocytes can become damaged and are then lost. But OPCs have this amazing ability to replace lost oligodendrocytes in a process known as remyelination. And this is a major area for regenerative medicine for the CNS. But what we found was that when we shine light onto these OPCs at a much lower dose, and the doses here I'm referring to are about half, which is 180 kilojoules, to 108 joules, that's about a third for what we shined light on the neurons and astrocytes, the OPCs were completely lost. So this said that the OPCs are more sensitive, but it also meant that we might be able to use them to actually screen for sources and solutions to phototoxicity. So the simple experiment we did to figure this out was that we just took 6 weld plates. We had normal media in them, which in our case was DMEM, double echoes modified eagle medium, and we had Sato in there as well. Sato is a serum-free supplement. It's similar to N2, if anyone of you are familiar with that. And when we shine light onto this media versus media that was kept in the dark, we placed cover slips of viable OPCs into media that was kept in the dark or exposed to light. And what we found was that the OPCs that were placed in media that was exposed to light all died. So this says that the primary source of toxicity is actually in the media. So we can now use OPCs to solve this. So we came up with Memo media, which is modified eagle medium for optogenetics. We screened for compounds that could react to light in normal DMEM, and this is how we came up with Memo. And this is what's part of the LiveLight package from Cell Guidance Systems. And when we place OPCs into Memo, what we found was that it increased the viability of the cells when we exposed them to light. Now, it didn't completely restore it, but it was an improvement. What we found next was that when we increased this dose of light twofold, the complete phototoxicity was restored. Now, this means that in this photoinert media, we are now getting phototoxicity again. And the simple reason for that is that the toxicity is occurring inside the cells. So this is where we generated supplements for optogenetic survival, SOS for short, And these are based on N2 and other serum-free supplements such as NS21 and B27, but with additional antioxidants and vitamins. Now, I'm showing you this image again, which is on the left of when we have OPCs in Memo, and as our serum-free supplement Sato, that when we shine light on them, they all died. But in the Memo media, which is in the middle panel, with SOS, we were able to preserve their viability. And if we increased the light dose up to 2,160 kilojoules per meter squared, which is 20 times the dose that completely destroyed the OPCs, we still have viability. So this is a staggering increase in the amount of viability we've managed to achieve with the media. Now, that's because there's two sources for phototoxicity. The first source is in the media because there's chemical reactions and photochemical reactions occur, so Memo blocks that from happening. The second source is inside the cells themselves, and that's what SOS prevents. Now, the one thing that you should bear in mind is that when you are growing cells day to day, they're in ambient oxygen, which is 21% or thereabout, which is a staggeringly high level of oxygen. So we are routinely using SOS as our normal day-to-day serum-free supplement, which is in keeping our cells much happier in what is a high level of oxygen in normal cell culture conditions. We also came up with Pneumo, which is neuronal media for optogenetics, which is based on Memo, but modified, as Sami was showing quite well, for the neurons. And when you combine Pneumo and SOS, you can shine light on neurons without any side effects. And we were also able to show that there is no loss in the astrocyte morphologies when we shine light on them. So essentially, we are now able to do optogenetic or live cell imaging or super resolution imaging or FACS sorting on these cells without any side effects or concerns for the effects of light. Now, one of my colleagues, Katrin Volbracht, was working on calcium imaging in OPCs, and anyone who's familiar with Führer 2AM calcium imaging would realize that you use low wavelengths of light of 340 and 380 nanometers. So what Katrin found consistently was that when she was doing calcium imaging for a period of about 80 minutes on OPCs, that in the middle panel, what you see here is that at the end of the calcium imaging experiment, there's an increase in background noise. So now this is being done in Ringer's media. Now, Ringer's media is essentially a photoinert buffer, which is used quite commonly for electrophysiology, but also for imaging. So we postulated that normal cell culture media actually loads cells with components that react to light. So to test this out, sorry, this is just the histograms to show the quantification of these background calcium stains. But if we place OPCs into Memo and SOS prior to imaging, and then we perform calcium imaging on them, what Katrin was able to show is that there was no major increase in background noise. So Katrin's calcium imaging was improved. Now, another colleague of mine, Abby Crawford, was very interested in isolating OPCs from adult mice, and these OPCs were genetically labeled to be green or red with GFP and RFP proteins. And when Abby did a flow cytometry, what she found was that she wasn't getting any of the cells. This is because she was using normal media, which is neurobasal and B27 in her case. But when Abby switched over to the Pneumo and SOS, what she found is she was able to eventually sort cells and keep them alive for 5 days and be able to study them and generate data. So to summarize, there is a dark side to light, and one needs to appreciate how much light is being exposed to cells, and the best way of doing that is to take into account light can be considered just like a drug. It's all about dose and exposure. All CNS cells are altered by light, as I've shown you here, but this probably extends to all cells in general will be altered by light. And the source of damage is in the media, but it's also in the cells. And by using LiveLight, you can extend the amount of light that cells can withstand up to about 20-fold. Now, this means that LiveLight is a cheap and economical means to actually do intense imaging or exposing cells to light without fancy optics or other hardware. So I just need to acknowledge all those that were involved in this work, which is was done in the laboratory of Raglan Hilder, Cara Doter, and all her lab members, such as Kimberly Evans, Morris Matthew, Catherine Volpracht, and Sylvia Gahu. I also need to thank Juan Barone and Yana Mukanoa in King's College London for their help in testing the media, and also those of Cambridge Enterprise, Rachel Atfield and Laura Donnelly, and Cell Guidance Systems, and Michael and his team. So I'm happy to take questions now.
Okay, thank you very much, John. That's an excellent presentation. So we have a facility for people participating in the webinar, people listening in, watching, to ask any questions they may have. So if you'd like to type those in, they'll appear here, and I'll read them out, and either John or Sami can answer the questions. But maybe I should start off by asking a fairly basic question, which is, where do you see this technology having the largest impact, and what kind of experiments are most affected at the moment, where people are really struggling because of problems with phototoxicity? So maybe I'll address that to John to start with.
Thanks, Michael. I think that live cell imaging, which Sami is doing, I think that's probably the first area that people will have problems with phototoxicity. Firstly, because microscopes and live cell imaging is an expanding technique now. It's in almost every laboratory. But the other aspect to it is that people don't take into account or appreciate just how damaging light is. And while they're only looking at one cell at a time, they may not realize that that cell is actually being affected, because the other population of cells around them may not. The next area will definitely be optogenetics, because that requires high levels of light in order to activate the different proteins. And it is impossible to do optogenetics with those levels of light and not expect to have any side effects. And after that, I think the main source of toxicity from light that people are going to be aware of is when they're not managing their media correctly in day-to-day cell culture.
Okay, that's great. Thank you very much, John. So for you, Sami, how would you say that using LiveLight has impacted on the output from your lab?
Yeah, I would probably echo a lot of the things that John had to say already. I think it's not all about static fluorescent imaging now. There are so many really amazing tools out there based on fluorescent proteins, or, as John was mentioning, optogenetics. I mean, there's a system for, it's called OptoPatch, developed by Adam Cohen, which is actually a fluorescence-based voltage monitor that is connected with a channel rhodopsin derivative that allows you to stimulate with light as well as record based on the emission of light. And both because of toxicity, required for the stimulation, but also for the sensitivity to pick up small changes in membrane potential, you really need a type of media to support these experiments. So I mentioned we're working with a lot of photoconvertible proteins, and now you can use these to really monitor any number of physiological processes. And a good example is calcium imaging with some of the GCamp, say, or some of the dyes. You want to be able to illuminate continuously, or at least semi-continuously, and if you do that, you're just going to blow up the cells. So LiveLight really helps in that aspect.
Okay, that's great. Thanks, Sami. So I've got a question here from Joe. I hope I'm pronouncing the name correctly. And Joe would like to ask if you have any experience on how these media work with other cell types. So I guess if both of you could address that, if you have any experience. Maybe John first.
Sure. I'm using Memo and SOS as a replacement for DMEM. The other cell types that I would use would still be CNS types of cells. There's microglia, but in terms of cell lines, Memo should be a complete replacement for DMEM for the imaging purposes, but I'm using SOS currently for myelinating cultures as a complete replacement for B27. So yeah, I'm continually using them, and I think you could probably use them as a direct replacement for any media that would normally be using DMEM, which would be the vast majority of cell lines.
Yeah, in terms of our experience, it's mainly been limited to primary neurons in culture, primary astrocytes and glia in culture, and we have used it a little bit with some immortal cell lines like hex cells. It works beautiful. It's sort of overkill for the hex cells because they can live through anything. And then we're also now starting to use it in some of our differentiations of human adult stem cells into mature cell types, including neurons and glia. So that's been our experience, and it works. It should work as a base for the IPS-based media as well, but we haven't really been there yet.
So you showed during your presentation, Sami, that the media LiveLight media was able to reduce the general background intensity that you were seeing on some of your preparations. Yeah. Is that one of the main advantages of using it with hex cells?
Yeah. In particular, we're looking at, we're using CRISPR-Cas9 technology to knock in fluorophores so that, you know, they're really only expressed at endogenous levels, and when you don't overexpress these fluorescent proteins, they're not really as bright as you might think. And so you really need anything that can enhance your signal-to-noise ratio, whether it's an EMCCD or you know, gain, or reducing the background is really going to help.
Okay, that's great. I don't see any of the questions coming in, and obviously due to the technical glitches that we had at the start, the number of people who've stayed on is relatively low. So I think that's probably a good point to end the webinar. I'd like to thank everybody who did stay on and persevere with us. It's great that you were able to do that, and we could share this this important technology with you. So the webinar should be recorded, hopefully, any technical glitches permitting, and that will be available for the foreseeable future. So I'd like just to end by thanking very much our two presenters, John Stockley and Sami Barmada, for really making some very interesting presentations about the media. And if anybody has any questions that they would like to direct to us, to our technical services, then all the information is available on the Cell Guidance Systems